Well, I'd like to welcome everyone to our Women Leadership Awards webinar this morning. We have a great group of speakers who are going to tell you about their science and careers. So we're very excited to have you join us for this exciting event. I also want to mention a couple of other things. The WMIC 2023 symposium will be in Prague, in the Czech Republic, from September 5th to 9th this year. And one really exciting new development is that PIs can register your trainees for free. The abstract deadline has been extended to May 1st to allow everyone to take advantage of this opportunity. So please think about registering your trainees. Also want to mention one other upcoming webinar on metabolic activity diffusion imaging. This will take place on April 25th, 2023. And so we really look forward to this event as well. But today we're going to start off with Veronica Clavio Jordan, who's going to be telling us about her career and science. Veronica obtained her doctoral degree from Arizona State University, where she began working on iron oxide-based nanoparticle contrast agents. She then obtained her postdoctoral training at the Southwestern Medical Center and further developed work in gadolinium-based zinc-sensitive MRI contrast agents. In 2018, she joined the Martino Center for Biomedical Imaging at Massachusetts General Hospital, where she's now an assistant professor. She's received a number of prestigious awards, including the NCI Transition Career Development Award to promote diversity in the biomedical sciences community. She also has received a Physician Scientist Award from MGH and is also chair of the ISMRM group in molecular and cellular imaging. So we're very excited to hear from her about her work and her career. So I'm going to stop sharing, and Veronica, you can go ahead and share.
Yep. All right. Thank you so much for the introduction. I'm truly grateful for the recognition, certainly. And I wanted to briefly tell you a little bit about my journey. So far, I was born and raised in La Paz, Bolivia, and decided to migrate to the US when I was 18 to go to school and seek a brighter future. So I moved to Arizona first, where I did my PhD in undergrad, then to Texas to pursue my postdoc, and finally settled in Massachusetts to join the Martino Center, where I'm establishing my cancer-focused imaging group. My research interests have largely encompassed using MRI to answer the many complex questions that treating and curing cancer poses. To do so, I set out to characterize the tumor microenvironment, and we know that the tumor microenvironment is comprised of parenchymal functional cells, immune cells, stromal cells, ECM components, a variety of cellular key players that dictate really the fate of the tumor, its growth, metastasis, response to treatment, evasion to immune attack, etc. So very much like an art piece, one needs to step back and see all the different individual colors and strokes to really appreciate the art as a whole. So because I'm an MR imaging scientist, I thought in cancer, how can we appreciate the complete picture by MRI? We need, and we reasoned, we need probes, MRI probes that are sensitive to each one of these components. So for example, to image parenchymal and functional cells, we can use zinc-sensitive probes. We can use ROS-sensitive probes to report on immune cell activity and infiltration. We can use collagen-targeted probes to report on fibrotic status in ECM. And I'll begin by showing how we've imaged function using zinc-sensitive probes. So what we found is that zinc is commonly co-secreted in secretory organs as a response to a stimulus. So for example, in the endocrine pancreas, we see that the endocrine pancreas secretes insulin as a response to an increase in blood glucose. The exocrine pancreas secretes digestive enzymes and bicarbonate for digestion as a response to cerulean or secretin stimulus. And we discovered that the prostate also responds to glucose stimulation and secretes prostatic proteins and citrate for fertilization. So during my time in Dean Sherry's group at my postdoc, we developed a library of gadolinium-based contrast agents that are sensitive to zinc, that upon binding to zinc, they form a complex with albumin, they form a large macromolecule, and that promotes a change in relaxivity, which translates to an increase in image intensity by T1 weighted MRI. And what we observed was that the prostate of fasted mice, if promoted a stimulus, in this case glucose, and also provided the gadolinium-based zinc probe, we can see that the prostate secretes large amounts of zinc, and we see a very significant enhancement in the gland. So because this is unknown and it was a discovery, we decided to try to understand how are we detecting or what are we actually seeing. And we used synchrotron radiation X-ray fluorescence to be able to image prostates that were not stimulated and that were stimulated, and actually get metal maps or elemental maps of these glands and try to see both zinc and gadolinium. So in this case, we take a non-stimulated prostate that received the gadolinium-based probe. We can focus on the region of the gland where we see these glands that are filled with secretions. With SR-XRF or X-ray fluorescence, we can delineate the membranes with a phosphorus map. We can also see the gadolinium distributed evenly in the extracellular and extraglandular spaces. And we see that zinc is actually found very confined inside the glands in the non-stimulated form. Now, if we look at the stimulated prostate with glucose, we look at the same region, we can see that by XRF, we delineate the membranes with phosphorus. We see the gadolinium distributed similarly in the extracellular glandular spaces. But in this case, zinc is actually moved from the intraglandular space to the outside, where it comes in contact with our gadolinium probe, and that's how we see enhancement or increase in T1 weighted signal. So we capitalized on the fact that zinc content is significantly lost in prostate cancer, and we leveraged that loss to detect prostate malignancy by the zinc secretion MRI paradigm. So here I'm showing healthy animals that are stimulated with glucose and receive the zinc probe, and we see nice enhancement in the prostate as a healthy prostate would. Conversely, on a tramp mouse, that's a transgenic mouse prostate that develops a spontaneous cancer, we can see that the gland itself doesn't secrete the same levels, so the enhancement is lower. We also are able to discriminate lesions that are starting to form in the gland, shown here on H&E and also shown here on the zinc and gadolinium maps. So then the arc of this work and on the work that I'm pursuing really rests on the idea that by selecting the appropriate secretagogue or stimulant, one can target the secretion in respective secretory organs. So here I'm showing a secretory cell that could be from any organ that's decorated with a zinc indicator, fluorescent indicator that only emits fluorescence when it binds to zinc. And so, for example, if we were to use secretin or cerulean, in this case, one would promote the secretion of zinc from the exocrine pancreas only. And we found that in human pancreatic cancer cells, this was the case, where we promoted the stimulus in vitro, we used the zinc fluorescent indicator, and sure enough, we can see the fluorescence increase over time. So we tested the ability to see zinc secretion in a PDAC or pancreatic ductal adenocarcinoma mouse model, and where tumor cells are implanted in the tail of the pancreas and allowed to grow for 1 and 2 weeks. When when we administer our gadolinium-based probe and stimulate the tumor-bearing mice with cerulean or saline, or healthy mice. In this case, I'm showing healthy on the top. The pancreas does not enhance if they either with saline or cerulean in the probe. However, when we have cancer present or the tumor-bearing animals showed significant pancreatic and tumor enhancement only in the stimulated groups. On the larger mass or 2-week PDACs, we see that the tumor-bearing pancreas also enhances, and the tumor doesn't do it to the same level as the 1-week tumors. So because of that, we separated the ROIs between tumor-bearing and the tumors and try to quantify their zinc secretory capacity. So we see that the animals that were a 1-week tumor exhibited a highest change in secretory activity in the pancreas, while 2-week tumor-bearing pancreas showed diminished zinc secretion. Additionally, the actual tumor cells showed increased zinc secretion while they were small, and at 2 weeks, their secretory capacity was lost. So this is likely because in the presence of a large mass, the pancreas itself becomes atrophied, so it does not respond to the secretagogue. We validated these findings with ex vivo histology or IHC, and clearly we were able to identify the transporters that actually are responsible for taking in and out and keeping the tight homeostasis of zinc content in these cells and these tissues. And what we see is that ZIP3 and ZIP4, which are import transporters, are significantly upregulated on early PDAC tumors. The exporter as well is upregulated, meaning that in cancer, the pancreas not only does it take more zinc from diet, but when stimulated, it also exports a lot more. So to compare that and to characterize these same zinc transporters in human PDAC, we observed actually the same profile where the quote unquote normal pancreas shows low expression of import and export transporters, and the PDAC lesions and cancer-associated inflammatory tissue around it shows increased transporter expression, highlighting this as a potential for this techniques to be used in the clinic and useful in the clinic. So changing gears to understand the role that immune cells play in cancer development and be able to see the complete picture as we're trying to do, we use ROS-sensitive probes. So here we used a probe developed by Eric Gale's group. His probe is an iron-based molecule that is rapidly oxidized from a low relaxivity iron 2 complex form to a high relaxivity iron 3 complex, only in the presence of ROS or reactive oxygen species. This probe is essentially MRI silent in its iron 2 form and renders high image contrast only in its iron 3 form, as it's shown in this phantom T1 weighted image. We've applied this probe to image inflammation in models of acute pancreatitis and to image NASH or non-alcoholic steatohepatitis development on a CDA half-fat dietary model. And we also looked at acetaminophen overdose as a model of drug injury in the liver. And what we find is that the main culprits of ROS that is detected by this probe are likely neutrophils and macrophages. And we saw that by the increased MPO or myeloperoxidase activity and how that correlates with signal intensity in the pancreas, as well as neutrophil infiltration in the liver. So because I'm interested in cancer imaging, we set out to use this iron probe to image the role that ROS may play in NASH to HCC progression. So here we fed animals a choline-deficient half-fat diet for 16 weeks, essentially observing the spectrum of disease that goes from steatohepatitis, fibrosis, cirrhosis, and it is considered that one of the main triggers of HCC carcinogenesis and progression is infiltration of myeloid cells, immature myeloid cells, and dysregulated production of cytokines as well. So in this model, after 16 weeks on the diet, we saw that 40% of mice progress to HCC and the rest don't and remain on a NASH background. And we quantify the neutrophil and macrophage infiltration because we know that that could be a correlation to our signal intensity. And what we found is that both neutrophils and macrophages are definitely more present and prevalent in NASH relative to healthy, but even more so are these animals are the animals that progress to HCC or hepatocellular carcinoma. So in this context, we should be able then to detect this with our ROS probe. And here we administered the iron probe and saw that the livers of animals who progressed from NASH to HCC had significantly increased signal intensity in the liver after receiving the probe, and that the animals that are only NASH showed decreased change in the signal intensity in their livers. This is represented here in these curves where the signal is elevated in the HCC relative to NASH. And so to us, this gives us a possibility to image longitudinally with this probe and predict NASH to HCC progression earlier at times when therapeutic interventions could be possible to stop such progression. And finally, to interrogate the role that ECM components have on carcinogenesis, we used probes sensitive to collagen in PDAC. Here, Peter Caravan's group developed a gadolinium-based peptide probe that binds to type 1 collagen. And with this probe, we were able to interrogate the fibrotic microenvironment in PDAC, where the collagen probe is retained in the fibrotic tumor, while gadolinium DOTA control does not retain and washes out quicker. And importantly, given that humans post-chemoradiation fibrosis is actually associated with longer ROS, overall survival, and disease-free survival, we treated PDAC mice with FOLFIRINOX, which is a common chemotherapy treatment in PDAC, and found that the collagen-targeted probe showed increased MRI signal and retention in the tumor compared to untreated groups, highlighting then the possibility to use this probe to assist in prognostication of disease. So in this way, we slowly begin to see the picture with probes designed to be sensitive to these different components. So we can interrogate function, we can interrogate immune cell infiltration and activity, ECM, but of course there are more aspects and components that need to be imaged, and therefore we need compounds that could be sensitive to these. So since science is definitely not done alone, I can't ignore and not acknowledge the help of my students, colleagues, collaborators who made all of this work possible, and of course the funding sources who support this work as well. And I can't, be remiss not to mention the role that mentors play in my career and in a young student's career. So in my PhD, Kevin Bennett, he was my mentor, and he formed me as a young scientist, and really gave me the confidence that I needed at the time as a young female, Hispanic female in academia. Dean Sherry and my postdoc, he fed my curiosity and always believed in me and continues to do so. And Peter Caravan, who continues to support me through his invaluable mentorship till now. So with that, I'd like to thank everybody, and thanks to the Society for this incredible recognition.
Thank you so much,
Mhm.
Veronica. That was a great presentation. We're going to
Thanks.
hold questions until the end for all three of our panelists today. So yeah, thank you so much, and we'll look forward to a question and answer session at the end of our program. All right, and so oops. So now I would like to introduce Naomi Matsura, who will be giving our next presentation. So Naomi received our inaugural Mid-Career Award this year. And Naomi is an associate professor in material science and engineering and biomedical engineering with a cross appointment in the Department of Medical Imaging at the University of Toronto. In her research program, she designs new translational theranostic biomaterials for the treatment of human disease. She's received a number of awards, including the Polanyi Prize in Physiology, Medicine, and Physics, which is an early researcher career award, and the Suffrage Science Award Naomi has served the WMIS and women in a number of different ways. She was a chair of the women group. She also serves on the board of trustees of WMIS. She was a program chair also for the 2020 World Molecular Imaging Congress. She also is a member of the WMIS DEI Advisory Committee and the Program Advisory Committee for the Nanomedicines Innovation Network. She also sits on the editorial board of Molecular Imaging and Biology. And she's had a particularly interesting career that we're hoping that she's going to tell us about a bit in combining science with dedication to fairness and equity, and has had some really unique experiences and contributions in this regard. So thank you so much for joining us, Naomi, and let me stop sharing so that you can share your slides.
So thank you, Kathy, and thank you, everybody. And I want to thank you so much for this honor of receiving this award. Today, I thought I'd do something a little bit different and talk a little bit more about this career trajectory and some of the choices I made in my life rather than focus primarily on the science. So I hope that is okay with you. So I'd like to start first with sort of this concept of not knowing what I wanted to do since I was very young. So I never knew what I wanted to do. I still don't really know what I wanted to do. All I knew when I was young was that I liked doing puzzles. I liked doing meticulous and careful tasks. If someone said something was hard and challenging to do, that meant I wanted to try it. I loved school when I was a kid. I never wanted to miss a day, even from kindergarten all the way to the end of high school. And but I also like things like drawing and painting and crafts and reading and sports. And if you gave me something that, if you told me that I had more responsibility, I'd like that kind of job. So if you said, oh, I had to do something and you're going to be responsible for something important, I wanted to do it. So what that meant is when I went to university, I had really no idea what I would be suited for. So I picked a program that I thought would give me the most options. So that, in my mind, was the engineering physics program at Queen's University in Kingston. I looked up what the description of this was, and it says this is a course designed to fit men for positions as physicists in research labs. And this is when it was introduced in 1919. So in some ways, except for the men part, it fits me quite well in terms of what I'm doing now. In the class, in the undergrad, there were 45 students or so and 3 women. I was the only Asian woman in this program, and there were no female professors. Although it was a very small program, it was quite prestigious at that time in that it had a very large focus on particle physics. We had one of the few Nobel Prize winners in Canada was our Professor Art MacDonald, who won it in physics in 2015. And so I was proceeding this program, very challenging, very time intensive. But 2 really important things in my personal life happened during this time. One was my brother was diagnosed with a non-Hodgkin's lymphoma for the 2nd time in first year university. And the 2nd university, my father, who was a really big supporter of education for me, which was quite rare in, as someone in my family, he was killed in a car accident as a passenger in 2nd year. So what this did for me was made me think that life is short. So this shaped my worldview, and how the choices I made after this was based on this idea that those opportunities I should take. If there's some opportunity for me, I should take it, because I don't know when that opportunity will go again, and I don't know when I'd have a second chance. So I didn't like to wait. So this is sort of this has infiltrated all my choices and how I conducted my life, which will maybe explain my bit circuitous and journey that I've had since then. So after my undergrad, I still didn't know what I wanted to do. So I continued with my master's in solid state physics. I published my first paper and on thin film nanostructures and the interactions of these films with ion beams. After that, I worked at Chalk River Labs, and this is a nuclear research facility in Northern Ontario. It's about 10 kilometers upstream from from the town that you lived in. So there I looked at the irradiation damage from neutron beams on zirconium pressure tubes. I had, published some papers. One of them were and a first author paper. And I also had a really great work-life balance. I would ski to work in the winter, and I'd bike and kayak to work in the summer. And it was a really idyllic environment to do work and live. So after my 1 year there, I was offered a full-time position, but I felt I wasn't ready. So from then, I decided, okay, I wanted to travel. I wanted to see the world. So I got a working holiday visa in Australia. So I went to Australia with a backpack, and I planned on, finding these casual labor jobs and travel around the country. Fortunately, the only thing I was qualified to do was really work in research. So I worked at CSIRO, which is a national research institute in Australia. And there I was making quantum dots and nanoparticles, similar to what I did with techniques I learned in my master's. I had a publication there as well. And then the rest of the time I backpacked to Australia and New Zealand, and really just used up all the money I made at this job, and then returned back to Canada when I just ran out of money. And so although these look like kind of different types of jobs, they were quite similar in the sense that they were all involved like hands-on experimentation and synthesis of nanostructures and characterization of surfaces. And really, I was always looking at how different radiation or energy interacted with these kinds of materials. So after that, I came back to Canada. Again, no idea what to do. This is sort of the theme of my life, I think. And what happened was my brother was dating a woman who had just finished her master's in medical biophysics at the University of Toronto under John Rowlands, who was in charge of the X-ray imaging research group at Sunnybrook. So he was looking for a technician, lab technician. So I went to him with my CV, and he looked at my CV and said, well, I think because you've done these papers and you've done all this research, I think you should do a PhD. And I said, there's no way I'm doing a PhD. I'm not interested in that at all. He goes, well, look, why don't you work with me this summer as a summer student and decide afterwards? So I said, okay. So at that time, I was commuting from my mother's farm to this hospital, and it was like a 2-hour commute during rush hour, but before and after rush hour was like 30 minutes. So that meant I had my working day was before 7 a.m. to after 7 p.m. So I show up before anyone showed up and stayed later just so I could avoid traffic. So what that meant in practice, that I had done a lot of work during the summer. So I was able to produce a paper at this end of the summer, and then this paper randomly became this one that was a runner-up for sort of the best paper in medical physics published in Canada that year. And really, it was because of the pedigree of the lab and the prestige of Sunnybrook that permitted this paper to be on the radar, to even be considered for this kind of award. But John Rowlands thought that I was this amazing emerging scientist because of this result. So anyway, so that kind of was both an honor, but also a little bit scary for myself. So what happened then is, after that, I decided indeed to do a PhD, and I actually went to do it in the material science and engineering department at the University of Toronto. It was a very hardcore solid state physics lab, and I found this to be an incredibly challenging experience, and it was tough. And I thought, because I'd worked in all these other labs and I was able to publish papers and I had little awards, that I was well suited for this. And in fact, I realized that I was supported by resources and people that facilitated this success. And if I didn't have that, I was nowhere. So anyway, after 5 years, I was able to cobble together my PhD. And again, sort of very similar to work I'd done previously, looking at making different kinds of nanostructures and looking at their interaction with electromagnetic radiation, and patterning these surfaces with new techniques that I developed. So as part of this PhD, I'd also, in the first bit, I was on the varsity rowing team, cross-country ski team, and at the very end of it, I coached some high school and varsity rowing and coached some cross country skiing. So, but during the PhD itself, I really was just focused on trying to get out of there. So after my PhD, I had no plans. And really, I had no plans because I didn't really think I would ever graduate. So people say, what are you going to do after you graduate? And I'm like, I'm not going to graduate. So there's really no point in thinking about it. But I did graduate. And then I thought, oh, what am I going to do? So it turned out I had 3 options presented. So one was to accept a postdoctoral position in Sunnybrook under John Rowlands. So that whole summer craziness and that award for that paper led him to think that he wanted to recruit me as a postdoctoral fellow. The other thing was an assistant professor position came up in that very department that I just graduated from. So what happened is there was no female professors, and the chair of the department said to me, he goes, we have to hire a female professor. I think you should apply. So after my PhD, I just had too much PTSD to even consider this. And so, so the third option that came up was to accept a volunteer position. And this is coming from context through my coaching that I had done for varsity teams at U of T at the university for sport and development NGO in Zambia. So, I agonized over these choices for quite a long time and asked everyone. Everyone said, you got to apply for this. This is the professor position. And a couple of people were saying, well, maybe you want to go to Africa. So in the end, I thought, well, the opportunity in Africa probably won't come back again. So I took that. So I worked for a year for an NGO called Right to Play. And it was really a life-changing experience, and probably not for the people I work with, but for me, in terms of learning about, the power and privilege I had, even though I didn't think I had it. The idea of prejudice or how we see other people, all the unseen workers who we never give credit to. And I think this experience really shaped what kind of influence I wanted to have later on when I returned to Canada. So after about a year and a half in Africa, so I worked for a year, traveled a bit, I came to Canada again with no jobs prospects or plan, which is kind of the theme of my life. But John Rowlands came back again. He kept thinking that I was this great student and great scientist. So he actually emailed me 2 days after he came back and said, I have this postdoc position. Do you want to join? I said, yes. And I ended up getting a 2-year fellowship with this program at Princess Margaret Hospital. And it was unique in that it was at this time supported by all these like physicians and biologists and physicists all working together to support the trainees who were all like postdoctoral fellows or grad students. And so this is my first sort of dive into this idea of real, cancer research as a holistic kind of field. And in 2007, as part of this program, I learned about WIMIC, and that was the first time I attended in Providence, Rhode Island. Another couple of things that happened was that I won this Polanyi Prize Award, which was actually, I don't think I was the most deserving, but I certainly appreciated the opportunity it afforded me through this recognition. And then the third thing that happened in 2007 is I was able to get a seed grant as a PI from the Ontario Institute of Cancer Research. And that was based on this topic called novel nanostructure assembly. So, and it was a small amount of money, but less than 50,000 US. And from that, I was able to hire a technician, and we put together these little different methods to make nanoparticles and coat them with different morphological layers for, payload release and protection structures, etc. So pretty soon after, I presented this to my colleagues in this program, it was clear that these were probably going to be very limited as clinical imaging agents, like they wouldn't clear, they don't degrade. So I thought after that I would have to think about new research directions. So at this time, John Rowlands, he had left Sunnybrook to become a scientific director at a different institute in Northern Ontario. So he had left Sunnybrook. But there was a big, bunch of senior ultrasound researchers at Sunnybrook. So, I started working with them, and then I got a grant from, again, the OICR to make bubbles and nanodroplets for cancer imaging. So this was enough money for me to buy equipment and have an operating grant for about 4 years. Also got a small concept award on an adjacent sort of topic from the DoD Breast Cancer Research Program. And what happened at that point, though, is I was still a postdoc. So it turned out that I wasn't allowed to hold these grants as a postdoc. So in 2008, I was promoted as a research associate, primarily so that I can continue to work on these projects. And then because I was a research associate, I was allowed to apply for a couple more grants. And then through the OICR, and then John Rowlands also put me as a co-PI on this large multi-institutional grant. And that funded me, paid my salary from 2010 to 2015. So in 2010, it turned out that I couldn't, as a research associate, I couldn't hold these grants. So I had to have a real position. So I got a 3-year junior scientist position at Sunnybrook and a lecturer position in medical biophysics. And so that's pretty much how I started my lab and started working on this area. This position allowed me to apply for grants for certain agency, but the problem was I wasn't able to independently supervise graduate students or postdoctoral fellows as a junior scientist. So through this, I was able to start this little program with microbubbles and nanobubbles and droplets and nanoparticles. I'm not going to talk at length on any of these things, but we were able to successfully work with scientists within Sunnybrook and in Toronto to make different kinds of ultrasound activated, light activated agents primarily for cancer. So I just thought, instead of going through this challenge of telling you about more of my research, I thought I would tell you about focus a little bit more on some of the struggles in the last few minutes. And so in 2013, I had this 3-year junior scientist position, and when my term was up, I was going to apply for an extension. But at that point, I received a letter before I put in this application saying that I was given 6 months to leave this institution. And that was not entirely unexpected, but I was surprised and quite devastated by this. But the issue is I still had funding for 2 more years of my salary and to support, I still had some funding to complete some research, but I had no position at that point. So what happened was John Rowlands, who had actually retired at the stage, he had heard that there was the chair of medical imaging was starting up a research program within the University of Toronto, and he was doing it alone. He had no one to help him. So I went to him and I asked him for a term-limited assistant professor position in medical imaging. And what that meant was that I would pay for my own salary, but I would help him in exchange for this position set up this course curriculum for the radiology residents in medical imaging, and to help him create a program to support radiologists to conduct independent research. So since he was at Sunnybrook, and since I already had a lab there and I had this new position, I just stayed in the same office and lab at Sunnybrook with this new position, which was confusing to everybody else in medical biophysics because I had just been let go, but I was somehow still there. And what this did is I spent the next 2 years, really only had 2 years of funding to pay for my salary. So I spent the next 2 years just trying to get as many grants as I could. So I got 7 grants, and some of them are really tiny to some larger ones over this next 2 years. And I tried to leverage the ability for me to get funding to get another job. So I would actually look for a job every month. I would put in a new application for a job every month. And to the point where I actually lost track of all the jobs I applied for. And in 2016, I got a job as a tenure track position in material science and engineering, which was also with biomedical engineering at the University of Toronto. And the irony is, this is the very job that I was supposed to apply for 12 years previously when I finished my PhD. So I went there in 2016 and I started again, started a new lab. And I got tenure in 2019. But this is the story that is not one that I tell. but this is the reality of it. And I feel that, this is behind everything that you see, there's kind of this kind of journey behind it. So I just wanted to let people know openly that this is sort of how that happened. So I just wanted to close with one more thing about being incredibly lucky during my career. And that is, and a lot of it is related to women in this network. There's some few pictures here. There's more, but this is what I was able to cull off the internet yesterday. And I just wanted to say, I attended my first women-related function 2014, and that's where I met Ann Menkins, who was a winner of the Leadership Prize last year. And she told me that I told her what I was doing in ultrasound. She said, "You have to talk to Kathy Farrar." So, and I said, "I can't talk to Kathy Farrar. I'm just like nobody." So Kathy talked to me for about an hour. And this was during the time I was having a lot of problems, and she really supported me in terms of trying to keep my spirits up to try to get this another position. And then I did, as Kathy mentioned, join the Women Leadership Committee, mostly from this meeting that really inspired me to try to do more for others the way other people have done for me. I made lifelong friends, and I just want to highlight, not only did I make amazing friendships and get a lot of support, I got numerous career opportunities in parallel thanks to this network. So from Julie Sutcliffe, Kathy, and Anna Wu recommended me for this other award. And this has led to downstream a whole bunch of other opportunities as well as scientific collaborations. So my time's up, so I want to end there, but I just want to, again, thank all of you for all your support and help. And I wouldn't have been able to do this without you. So thank you so much.
Thank you so much. That was a really inspiring talk. And yeah, thank you so much for sharing your journey and your experiences. It is invaluable to hear about everyone's journey and experiences, I think. So yeah, thank you so much. And again, we're going to hold questions until the end of our hour. And so we'll move to our last speaker. And And yeah, so it is wonderful now to welcome Anna Wu, who won the Women Leadership Award in this last year. Anna will be talking to us also about her journey and her science. She's the professor and chair of the Department of Immunology and Theranostics and co-director of the Center for Theranostic Studies at the Beckman Research of the City of Hope. She previously served as a professor and vice chair in the Department of Molecular and Medical Pharmacology at UCLA. And she was also director of the Cancer Molecular Imaging Program, also within UCLA and the Crump Institute. So Anna has worn a lot of different hats. She's a past chair of the California Breast Cancer Research Council. She was a fellow and past president of the World Molecular Imaging Society, and also received the gold medal from the World Molecular Imaging Society. She's a fellow of the Society of Nuclear Medicine and Molecular Imaging. She has done a huge amount of work in engineered antibodies and proteins for targeting and imaging. She's also a co-founder of Imaginab, which is translating radio-labeled antibodies for clinical use. So Anna, thank you so much for joining us, and congratulations on the Leadership Award, and as well as other honors. So I'm going to stop sharing and let Anna share.
Thank you so much. It really is an incredible honor to be recognized by your peers, and I'm really very grateful for receiving the Leadership Award from the Women, which is a group near and dear to my heart. Kathy was our founder, but I was one of the original members of the group, and continue to be active in the group. And I have to apologize in advance. I have a cold, so I'm a little froggy, and I hope I won't be coughing, but I will nonetheless forge on with this talk. So the title of my talk is Piecing Together a Life in Science. How did I get to CD8 immunopet? Because none of this existed when I started my career in science. But as I look back, there were a lot of threads that came together so that when I finally arrived at the idea, it actually made a lot of sense. And so I'd like to take you through that journey. And first I have disclosures. These are my financial disclosures. Of course, the relevant one is that I'm the founder and a current board member, shareholder, and consultant to ImagineAb. But first I want to go back and let's have a look at where all of these pieces came from. So unlike Naomi, I actually was born a nerd. My father was an organic chemist, a polymer chemist. So I grew up where we discussed things like the structure of glucose at the dinner table. So I was born in Waynesboro, Virginia. My father worked for DuPont on nylon, better living through chemistry. And then when I was 5, we moved to upstate New York, where my dad worked for GE Silicone Products Division, working on that silicone RTV that we use every day to caulk our bathtubs. So, he was a polymer chemist. And actually, I think an eye-opening experience for me was in junior high. I read this book, Isaac Asimov's Life and Energy, where he really describes how all of biology was based on biochemistry, based on chemistry, how every living thing on the planet comes from energy from the sun. So this was just fascinating to me, that living things run on very simple laws of chemistry, but how complex it was. And the other thing that happened while I was growing up was I was very fortunate to get a summer fellowship to work in a lab after the 10th grade. I worked with Leo Grady, and he was studying similarities between Bacillus subtilis and its bacteriophage, PBSX, and using reassociation kinetics. So this was long before DNA sequencing was invented. And the important things here are it reinforced my interest in DNA, but also provided early exposure to radioisotopes, because we were doing tritium C14 labeled DNA in order to track how they were reassociating. So at 16, I was actually too young to even have a film badge. You're supposed to be 18 to be, a radiation worker. But he did the handling of the stock, and then I would work with the DNA. I went to college then, and then my fascination with DNA continued, but antibodies sneak in. So my senior thesis was, I actually had the privilege in college of working with Matt Meselson of the famous Meselson stall experiment, where they proved the semi-conservative replication of DNA using isotopically labeled DNA molecules. And this is his Model E ultracentrifuge on which he did those studies. And I actually got to use his Model E centrifuge. So I was interested in DNA. So I did a senior thesis on DNA in Drosophila. But in my biochemistry class, one subject that caught my eye was this great mystery of antibody diversity. Because it turns out people like Lee Hood were sequencing antibodies and discovering this incredible variation in the protein sequences of these proteins that recognize, great diversity of different pathogens, et cetera. And it's been estimated that in the naive human repertoire, there could be up to 10 to the 12th different antibody sequences. And there's no way there's enough DNA in the genome to encode all of those proteins. In fact, it exceeds the coding capacity of the human genome by 4 orders of magnitude. So the big question was, how do you generate this diversity? And so the discovery of gene rearrangement to generate antibody diversity was just, it was heresy at the time. But it really was just a fascinating story that the DNA that we thought was immutable actually was getting routinely rearranged, So that caught my eye. It just sort of stuck as a very fascinating story. So I went on to grad school, and by then, fortunately, DNA sequencing had been invented. And so you can see through grad school and postdoc, I was interested in DNA's 4 letters. Yeah, we can encode proteins with them, but there's also a lot more information in there. RNA polymerases have to know where to start and stop. We have recombination. We have all sorts of other interesting things going on with the DNA. And so I was interested in proteins interacting and recognizing DNA. So one question was, how does RNA polymerase know when to stop? That was the subject of my PhD thesis. And what we and others found was you typically find a region of dyad symmetry that's GC rich, so it's very stable, followed by a AT rich sequence in the DNA. And what happens is when that is transcribed into RNA, you can form these hairpin structures. And although the details of the mechanism aren't known, it's thought to have a role of popping the RNA off of the DNA template and terminating transcription. So that was really cool. But the other interesting thing is that I actually ended up not in a biology department, but in the molecular biophysics and biochemistry department. And it's really stupid why I ended up there. It's because I was a biochemistry major in college, And I was afraid to take the GRE in either biology or chemistry because I didn't have enough biology and I didn't have enough chemistry. And this department did not require the subject matter GRE. So that's why I ended up there. But what it meant was I had to take the core biophysics course. So I had to learn about nanosecond fluorescence spectroscopy. I had to learn about NMR. I had to learn about X-ray crystallography, which included Fourier transforms and inverse space and all that stuff, all applied to macromolecules. So I've highlighted in blue words that kind of I somehow kept in mind and you should keep in mind. It's also where I met my future husband, Jeff. So that's a very important part of my story. So on to postdocs. And again, I stayed at Yale and continued to work on proteins that do interesting things to DNA. So worked with Charles Radding on REC-A protein, and it's involved in homologous recombination of DNA. So again, DNA not being immutable, but rather DNA being able to exchange, rearrange. And that was followed by a postdoc then at UCSF with Keith Yamamoto, cloning the glucocorticoid receptor gene. So the glucocorticoid receptor is a steroid hormone receptor that is a transcription binder. So it binds to DNA sequences. Again, how do these proteins recognize DNA? So I won't go into gory detail. The stint at UCSF was important because I wanted to move into mammalian cell genetics and molecular biology. And I learned techniques that were state of the art at the time, Southern blots, Northerns, Westerns, but I never did clone the gene for the glucocorticoid receptor. A different postdoc in the lab got it. And so I found myself at the end of these postdocs with no project to take to my next position. So I was sort of faced with what you've heard up from Naomi, what do I do now? Well, I was lucky in that I was able to find a position at the City of Hope, lucky in many, many ways, because this was my introduction to translational research. People were starting to recognize that the basic science that we were learning, the molecular biology that we were learning, that this knowledge could actually be useful. And so I have to tell you a brief story about how we used to diagnose lymphoma and how we diagnose it, well, not even now, but how it was evolving at the time. And so I actually had a joint appointment, not just in molecular biochemistry, but also in the anatomic pathology department in the hospital. And Henry Rappaport here is like the giant of classical lymphoma classification. In fact, they call it the Rappaport classification. And he mastered the field of being able to look on a slide and just to look at the cells and the tissue pattern and the morphology of the cells, the amount of size, cytoplasm, the shape of the nuclei, and be able to classify lymphomas in meaningful ways that impacted treatment. So that was the standard, but what was emerging was immunological classification of lymphomas. For me, what this meant was if you have a lymphoma malignancy, it's clonal. So normally in humans, there are two different kinds of light chains in your IgG antibodies, either kappas or lambdas. It's about a 2-to-1 ratio of kappa to lambda. But if there is a monoclonal malignancy growing out, it's either going to be all kappa or all lambda. So they would stain the patient specimens with antibodies such as kappa and lambda. And if you saw something like this, where it was all kappa and not lambda, they could clearly say this is malignancy, this is a clonal proliferation. So through that process, I learned all about hematopoietic differentiation, cell surface markers, CD antigens, they have markers for B cells, T cells, macrophages, et cetera. And I started to learn the language of these cell surface markers. But what I was doing was moving on to molecular diagnostics. So what we did was to take the patient's samples, extract the DNA, and do Southern blots to look for gene rearrangements, either in the antibody or the T cell receptor genes. Because again, if you saw a precise band, that was an indication of a clonal expansion, and that told you that you had a malignant process. So this is one example of the kind of work that we did with those Southern blots. And for me, this was really eye-opening that we actually could use these techniques to diagnose cancer in patients. At the same time, what was happening in City of Hope was a budding radioimmunotherapy program. And I have to take a slight detour again to mention Art Riggs, who passed away a year ago, very sadly, but a true pioneer. He came up with synthetic genes, including somatostatin and insulin. He also came up with recombinant proteins, such as antibodies. In fact, Shamili Kabili, the postdoc in his lab, is the Kabili of the Kabili patent on recombinant antibody therapeutics. So he worked on recombinant proteins, and then Jack Shively worked on antibodies that recognize CEA, carcinoembryonic antigen. And these antibodies were highly specific for colon cancer, but had no biological effect. So they were arming them with radioisotopes in collaboration with a surgeon and using this as a potential targeted therapeutic. So I actually, and I think because these concepts resonated with me, I joined that program and added further engineering to that process. A postdoc in ArtSci had made this chimeric antibody where he had rearranged to put the mouse variable regions onto a human backbone. And they were labeling this with indium-111 to image in patients, and then labeling it with yttrium-90 to treat patients. So here is an early example of theranostics where we would image first with a molecularly specific probe, and then follow that up with a targeted treatment. And then my role, as many of you know, has been to further take engineering as a way to improve antibodies for targeted radionuclide delivery. So we further made humanized or fully human antibodies. We worked a lot on developing engineered antibody fragments, such as mini-bodies and diabodies that target fast and clear fast. So they're actually optimized for imaging as well as therapy. And importantly, they're biologically inert. They're just vehicles for carrying the radionuclide to the tissue of interest. And we introduced changes in the protein sequence, such as cysteine residues that enabled site-specific conjugation. So here is an early paper of ours, actually our first mini-body paper showing it was labeled anti-CEA mini-body labeled with iodine-123. So these are planar images. They would actually sneak into the clinical imaging department, nuclear medicine department after hours, and put the mice under the clinical scanners to obtain these images to show the tumor targeting, because we had no small animal scanners at the time. So then I made a trip across town to UCLA, where first as a sabbatical with an amazing group of collaborators, Mike Phelps, Sam Gambier, Simon Cherry, Arian Hajwano, and this was the beginning of molecular imaging at UCLA and elsewhere, and was tasked with, first of all, imaging gene expression very directly. Can we use radio-labeled antisense oligonucleotides to measure the presence or absence of specific mRNAs in cells? And then what I brought over was the fact that I had these engineered antibody fragments that targeted fast, cleared fast, and would be perfect for PET. So this actually made a lot of sense to me. I actually embraced this non-invasive imaging of living subjects because it was biophysics, and we were still talking about inverse space and Fourier transforms. So it actually was very easy for me to embrace this concept of doing these, three-dimensional images. So I joined the faculty at UCLA in 2002. As you know, I moved back to City of Hope, as you may know, 5 years ago, but that was a very productive time. But there were things that were not so productive. So again, I wanted to point out that this first project went nowhere. And after 1 year, I told Mike that this is not a one-year project, it's a 20-year project. And I think it's 20 years later, and we still don't have a really good direct way to do this. A lot of indirect ways, but anyway, sometimes projects don't pan out, but you hopefully have 2 or 3 things going and can move on. So as many of you know, we applied this to imaging tumor markers. So here's just a spectrum of some of the cell surface markers in cancer that we engineered antibodies against and labeled with a variety of positron emitting radioisotopes for PET imaging. But how did I get into immunology and imaging immune responses? This was inspired at UCLA by Tony Rivas, who was one of the melanoma immunotherapists at the time, developing ipilimumab, basically anti-CTLA-4 treatment in melanoma patients. And he was observing a process called pseudoprogression, where patients might look worse at first on treatment. Their tumors would get larger, they get even hotter on FDG PET, but if you waited long enough, these patients actually did fine. They eventually, their tumors would disappear and they would do very, very well. And so we were having these weekly molecular imaging meetings, and he asked me at one point, how can we distinguish tumor cells from activated immune cells? And he also told me the cytotoxic T cell is the drug. And so we put things together, and I had remembered from my earliest work at City of Hope, hematopoiesis, the fact you have all these lineages, and that there were all these CD markers on the cell surfaces and corresponding antibodies. And those cytotoxic T cells, of course, are marked by the CD8 biomarker. So we tackled that first in mice. Very talented postdoc Richard Tavares, who developed used a rat anti-mouse CD8 antibody, developed an engineered antibody fragment that we labeled with zirconium, that we could map the immune system and immune responses in mice. To make a long story short, this is our workhorse on the mouse side, but along the way, I also co-founded ImagineAb, which developed the clinical agent shown here. Just a really brief overview of just a huge amount of work by a lot of people shows you that this concept of imaging those CD8 T cells as a marker of immune response is really quite broadly applicable. So with collaborators, we've looked at checkpoint inhibitor therapy, adoptive cell transfer, combination therapies, including a project with Kathy Ferrara, oncolytic viruses, bispecific antibodies, and most recently, radiation oncology resident in our lab has been working on looking at radiation and therapy combined with immunotherapy, and how does that trigger immune responses, as well as non-oncologic applications such as stem cell transplantation and graft versus host disease. So really potentially very broad applications. For me, a thrill was to be able to see this happen in patients. And this, of course, was done by ImagineAb, which did the clinical development and translation. So this is the phase 1 paper that appeared last year. The first 15 patients imaged with CD8 immunopet, showing the expected clearance to the liver, but also in all of these patients, spleen, bone marrow, lymph nodes. And in 2/3 of the patients, we were able to see tumor uptake of CD8 radio tracer. And what we're doing now is the phase 2 study, which involves patients who are scheduled to go on to standard of care checkpoint inhibitor therapy with a baseline CD8 immunopet scan and an early on-treatment CD8 immunopet scan to see, is there a change, is there an influx of those cytotoxic T cells in response to immunotherapy, which would give us an early indication that it's working. So here are some examples from that phase 2, a non-small cell lung cancer patient pre-treatment scan, on-treatment scan. Here are the corresponding biopsies, and you can see the increase in signal as well as the CD8 cells confirmed by immunohistochemistry. Likewise, here's an example in renal cell cancer pre-treatment versus on-treatment, and again, an increase in the infiltrating CD8 T cells. And it looks like there are some good correlations coming out of this. This is probably the best correlation, but you also have to bear in mind that the limitations of what we are kind of using as our gold standard, which is that CD8 immunohistochemistry, because the expression can be very heterogeneous, and we are of course limited by sampling what you can sample with a biopsy. And finally, just one example of the broader potential of this work, which is that it's not just about oncology and immunotherapy. This is work that Simon Davis and Negar Ahmedvari at UC Davis did, looking at infectious disease, in particular patients recovering from COVID-19. So here they found in a series of 6 or 8 patients at the image that COVID-19 patients scanned 8 weeks after their symptoms have resolved still have highly activated immune systems, especially in the T cells in the bone marrow, compared to people who have not been infected. So it just kind of opens the door to all kinds of questions of what's going on during COVID, what's going on during recovery, what's going on in long COVID, many exciting paths to explore. So I hope in this talk I've shown you how all these little bits and pieces of things that I picked up throughout my high school, college, graduate, postdoc careers kind of all came together to make this beautiful story of a whole body image, you know, and now you see the full quilt that is the product of all of these pieces. So the most important people in this work are my lab. So, and this has always been one of my favorite pictures. This is our group in 2015 at WMIC. Richard is the intact antibody. I'm the mini body. Amanda is a single chain FV. Kirsten and Wenting are a diabody, but and a guest appearance by Kirsten's mom. But my group at City of Hope originally, team at UCLA more recently, following my move back to City of Hope. This is our joint Wu and Zetlitz lab photo. I don't have room. I don't want to fill you with slides of pictures of all of my collaborators. I've highlighted some of the collaborators and mentors, but you know, at City of Hope, UCLA, Stanford, and many others. And of course, Team ImagineAb, who really made the clinical translation happen. And then finally, of course, this is a slide that was missing from my gold medal talk in Miami. They left out my family, my dad, my husband, But I want to leave you, well, just point out, these are my kids, Jeremy, Kelsey, and Lizzie. This is why I have a cold. Liliana turned 3 yesterday, but I was babysitting while her baby brother was being born, and she brought home a cold from preschool. Now I've got her, she shared it with me. This is Julian. He is now all of 16 days old. But I do want to leave you, before I go to the take-home messages, this is Kathy, my best friend from college, who made that beautiful quilt. Do what you love. And what I think you might find is that your head may think you're going this way, but your heart and your feet may be going in a different direction. And that's probably the direction you should really be going in, because that's where you actually will apply the most effort and be the most successful. As Naomi said, there's a lot to be said for being luck, being in the right place at the right time, but then you've got to seize those opportunities. I think it's important to be a perpetual student, because you can see it was not a straight path from DNA structure to CD8 immunopet, but I think it's important to constantly be open and learning, but patience and persistence helps. This took decades, to get to this point. And again, embrace change, because everything I'm doing now, none of it existed, remotely when I started my career in science. Science is continually evolving very quickly. So I will stop there and thank you again for honoring me, and thank you all for listening.
Thank you so much, Anna. Yeah, all very inspiring talks. Yeah, that was really a wonderful, wonderful exploration of your journey, and so we really thank you for that. We don't have a lot of questions right now that I see in the chat or the question and answer. We do have a comment, particularly thanking Naomi for sharing her story, but I think we appreciate all of your stories, that's for sure. So maybe I'll ask a couple of questions just to get us started and see whether or not it will spark others. But yeah, maybe Veronica, I would start with you if you wanted to say something about the challenges of starting your lab and what it's like to, in this day and age and with COVID and all the challenges that we face, what it's been like to get a lab started.
Yeah. So, as I was talking earlier amongst you guys, but I think all of us have gone through the similar things in terms of COVID. So, that was definitely something that impacted everybody. But for me, it was also challenging because I was starting. Funding got delayed. So things that were supposed to happen earlier couldn't, had to get rid of transgenic mice, and it just obviously a big setback. Another challenge that, I wanted to highlight, as a early investigator, also early, young mom. I just had a baby, not just, but baby a year and a half ago, my first daughter. And it is, it's a beautiful journey. But navigating the world and the life of an academic mom can be difficult and a little daunting sometimes. So I don't have, hindsight of like, oh, this is how things should be done so that you succeed. I will tell you when that time comes. But so that's been, the challenge of raising having a family, raising a daughter, all the things that come with, childcare and travel, going to conferences, having all the networking opportunities that we should, and disseminate our science and our ideas. But that means I had to go to AACR and bring my pump with me, and it's hard. Yeah, it was definitely an experience.
Thank you so much. Yeah, and please, Anna and Naomi, please feel free to jump in here at any point if you want to follow up as well. But
Well, I'd like to just sort of follow up on just the challenges of having kids and trying to have an academic career. Because I mean, some things have changed and gotten better, but some things haven't. And for me, I think I was fortunate to go to City of Hope as my first, independent job, because it was the whole process of getting promoted or tenure was much fuzzier. It took me 11 years to get promoted. If I had been in a full-blown tenure track, assistant professor position back then, I would have been out, not up. I think it's gotten better now, because now they'll stop the tenure clock for a year. that helps a bit, but it's still not easy. And what you said about travel, so important, to travel and to present your work and network. And, you know, at times I had a very patient husband. At times I would dump him with 3 small kids so that I could go to a conference. And he supported me in that. And it is really important, but it's really, really hard, you know, if you're a young mother, you know, if you've got young children at home. So I don't, even now looking back, I don't think there's any right answers. I don't think there's any best time. I think you just do it, and then you make a lot of compromises, and you find help everywhere you can find it, because it's absolutely worth it in the end. But it is, you know, you have no time for yourself. You just have to work, you have to take care of your family, and you just have to, that's your life for quite some years, but it's worth it.
Yeah, yeah, thank you for sharing those things. And I think even on the other end too, I know I've been on the other end of the sandwich and taking care of parents and having other, the other family responsibilities that fall on you as you live your life. And we all have, stories of the challenges of balancing all of those pieces. There are several other just comments in the chat now. Beautiful stories. Thank you all so much for sharing, and thank you so much, and thank you panels for sharing great stories. So yeah, I think there is a lot of appreciation for you guys for it, and it's not easy, I think, to share many of these aspects of your life. Um, yeah. Naomi, do you want to add anything else about, where you see your future going and things that, next challenges, what do you most want to do next?
I think in some ways, I think even this reflection has been really interesting to me in that, you have to be really objective about what we consider success and failure. Because I think it's exactly so, you could land on either side, and you can't be invested in either of those recognitions, whether people think you're a failure, whether you're a success. I think so for myself, I would like to mentor people. I think I really want to focus on my students and people to achieve things in a way that I haven't been able to do. I certainly have want to continue learning. I'm fascinated about, immunotherapies, a lot of the new technologies, new things that I didn't learn. So I want to spend a bit of time looking at that. And I'm lucky, like I have a job that only, I guess, I think probably priests or royalty had, a couple 100 years ago, 100 years ago. And so I can go in and learn for fun and interact with these amazing people who inspire me all the time. And I sometimes forget that. And so for me, I would like to continue learning, but I'd like to ensure that the next people I see don't necessarily go through the struggles that maybe Anna went through and I went through, and maybe Veronica's gone through. And so, I mean, and try to see if there's a way we can curve it so it makes it more equitable and easier for people to just focus on the science. And I think a lot of the times, even now, it's not that way. We're still focused a lot on barriers to even getting into the lab or getting to the point where we can do work. So, but, I certainly am, we still have a lot of projects going on in the background. So, but I recognize that this time with, students and, just the times we're living in now, it's pretty hard for people. And I find that a lot of the students that I have are having a lot of struggles in general. So I'd like to be able to support that and have a system where, they don't have to worry as much about these peripheral things, rather than, focusing on the science. So that's kind of what I'm thinking about right now.
Okay. Maybe one question for Anna too, and then we, after this, we'll probably go to the social lounge for about 10 minutes with a group. But Anna, I mean, you're kind of this role model for having developed the ability to image immune cells and actually make this happen in patients. When we see this next opportunity with diagnostics and with the ability to not just image, but also treat, what are your thoughts? What excites you most in that next step and realm?
Um, well, I think, that we've come full circle because as you can see, we were doing this 20 or 30 years ago at City of Hope. We just didn't have a buzzword for it. But there were so many details that had to be improved along the way. Isotope supply, overcoming people's fear of radiation. I mean, that always was a huge barrier to be, talking about injecting radiation into people. There's huge fear out there. And, it's just taken a long time for the whole field to progress. So it's not just us. I mean, it really even started with radium-223 approval by the FDA. I'm thrilled that we're now revisiting this concept because it makes a lot of sense to me. I love radioisotopes because you can find them and you can count them. You can image them and you can count them and you can do therapy. But also importantly, I've always liked radionuclides because they have a half-life and they go away with a very defined decay rate. So I think, it kind of appeals to the budding biophysicist in me. So this is what I mean by following, where your heart and your feet go, as opposed to where your head thinks you should be going. Because again, it's back to the laws of physics, the laws of chemistry. But we have this really cool set of agents, radioisotopes, that with a lot of different interesting properties. And I'm very excited that so many theranostics compounds and programs are now, finally blossoming on the radar as viable therapies. But I tell you, the most interesting question to me is the intersection between radiation therapy and immunotherapy, which has been kind of burbling along for a long time. But I think that having these targeted radionuclide therapies gives us yet another tool to figure out how to enhance immune responses, because low dose radiation does enhance immunity. So I think there's still a lot to be learned about how to use these therapies together. So it's a very exciting time.
Awesome. Yeah, it is a very exciting time. Yeah, and it does remind you of how it really takes addressing every detail of these compounds, and how challenging and how long it does take to create something that seemed like it happened overnight, but really is it has taken 20 years or more to get to a point. Well, so thank you all again. And, and I'd like also for anyone listening, we are still seeking nominations for the next set of awards. And again, Veronica received our Rising Star Award, Naomi, our Mid-Career Award, and Anna, the Leadership Award. And in all three cases, we welcome your nominations for this next group of awards for this next year. So please do think about this and think of others who are, we do have so many wonderful members of our community please do think about this. And we will be now going to the social lounge for a few moments. So please join us there if you have a moment. So thank you so much.